Okay, so can everyone see the slides? Hello,
Yes,
am I
sir.
audible? Okay. Okay, so let us start our class today. And before we start, so let us briefly look into what we have been studying in our last class. So we have completed the glycerol production, and we looked into the different pathways how we can produce the glycerol, and then we have looked into the what is the the growth curve, how the glycerol is produced along with the production of other intermediates, so like the ethanol, ethanol, lactate, acetate, or some of the byproducts that can be produced during the glycerol fermentation process, and the rate of their production. Okay, so some of the intermediates like the lactate or the ethanol, they might be used, can be used, these byproducts can be used for the production of the glycerol. So the once they are produced at some concentration during the fermentation process, they may on later stage be used up for the production of the glycerol. So similar, so those type of intermediates or byproducts are being produced for the certain period of time, like you can see in this particular graph, the production of the lactate, which was produced until the 40 hours, and then it was utilized as a carbon source for the production of the glycerol. So we have looked into this, the growth kinetic step, like how the production of the glycerol or the the concentration of the intermediate, especially the ethanol, lactate, and acetates, are changing with respect to the fermentation time. Next, we look into so given a we look in the process flow diagram. So given a biomass source to you, how you can process those biomass source to obtain the mixed sugars, which is nothing but the glucose derivatives, and those glucose containing media are inoculated with microorganisms, especially the microorganisms suitable for the production of the glycerol. And then we carry out the fermentations, and we have seen what is the conditions at which you have to ferment. So we have seen what are the the nutrients that are required, like for example in this case we required some of the nutrients such as the biotin and other urea, nitrogen source, and so on. So when we add the adequate growth media components, we optimize their concentrations, and then we carry out the fermentation under the aerobic conditions. So we have to provide the sterilized air. So sterilized air is nothing but a air that is being passed through the filter to remove any contaminants that are present in it. Okay, and we make sure that we have a highly aerobic conditions, so that we have higher production of the glycerol. And after the glycerol is being produced, the next step that comes is the downstream processing. And we have looked into there are several strategies to carry out, like first step being the filtrations to remove. So filtration will be carried out to remove, the biomass that is being present in the after the fermentations, followed by the evaporations. Then we have the vacuum carrier distillations to extract the the glycerol, and then that extracted glycerol is passed through the activated carbon to carry out the decolorizations, followed by the filtration, and then followed by concentrations to obtain a very high concentration of the glycerol. So generally the concentration reaches to as 99.99%, and so on. Okay, so this is the overall flow chart, how the glycerol productions takes place based on the fermentation approach. Okay, and there are several parameters that need to be monitored. So we have seen what are those parameters. Like for the fermentations, we have to tune the temperature, the aeration, and the concentration of the phosphate and the nitrogen has to be monitored during the fermentation process, and the concentration of the sugar. So as we know that the concentration to this particular graph, if we remember, this shows that the concentration of sugar is decreasing with the time. So that means after, for example, the 72 hours, when the concentration of sugar is very low or minimal, and then the fermentation will almost stop, because there will be not enough carbon source that can be converted to the glycerol, right? So we have to always make sure that we keep a track on how the sugar concentration is changing. And generally, if you are carrying out a fed batch process, okay, So in that case, we make sure that we add, the once that sugar concentration dips down, we add the adequate concentration of the sugar so that that fermentation step process still continues. And also the osmotic pressure. So osmotic pressure can be monitored depending upon how the, how much amount of glycerol is produced. So because here you can see that there are different components being produced. Like for example, this particular is shows the production of the glycerol. So as the concentration of the glycerol is going to increase, and as the concentration of the glucose is going to decrease, what will happen? The glycerol that is there will lead to the presence of osmotic formation of osmotic pressures across the cell membranes that are there. So to prevent such the formation of such type of osmotic pressure, what is happening? We need to remove the glycerol from the growth media, or as it is being produced, so that there is not enough high osmotic pressure that it may lead to the rupture of the cells or killing of the cells. So these things, or these parameters, needs to be monitored while we are doing the fermentation of the glycerol. Along with that, the other things like the temperature has to be monitored. So temperature, we have seen, depending upon the microorganisms, so there are different temperature ranges that needs to be done. And also the the aeration. So as I told you, this aerobic process, so we have to have the optimum DO level, and this concentration of DO is also depends upon what are the components that are you are adding, and the temperature at which you are carrying out your the culturing your fermentation process. Because the solubility of the oxygen in the gas, or solubility of the oxygen in the growth media, will be a function of both temperature as well as what are the components that are, or the salts that are being present. Okay, so that is why these two parameters are also important. And then we have seen that the recovery processes. So if you go with the traditional recovery process for distillations, generally it is having the 50% recovery. So therefore a specialized distillation based on the carrier method is used to carry out to improve the recovery. And by by the additional steps of, suppose, the ion exchange resins and the ion exchange chromatography, we it was found that that non-ionic impurities, basically the salts or the other impurities that are present, those can be efficiently removed and can be used efficiently removed, and we can improve the glycerol utilization of the production. And finally, we have seen that after the production of the glycerol, what are the applications of this? So you can use this glycerol to produce different amino acids, and we all know the amino acids is being used as a supplement, as a food material, especially for the bodybuilding and other applications. Other process also, it is used to make sure that your food have the adequate source of the amino acids or nitrogen sources, so that there is no the limitation of the growth elements that are required. Okay, so glycerol is widely used to produce different amino acids like tyrosine, the serine, and also the lactates. Okay, so this has been the overview of what we have studied for the glycerol. Next, let us move to the next topic, which is a very interesting topic. It's a single cell protein and the mushrooms. I'm sure everyone had have some time or ever have had the have eaten the mushrooms. And the single cell proteins. So single cell proteins are very interesting. Like as the name suggests, they are singular or single cells, and they are very rich in the protein content. Okay, and because of this, they are used in different applications, especially in the food additives. And if you see, for example, if you think of in the space missions. Okay, so in the space mission, when the astronauts go, for example, for the space missions, they need to have the food, right, for their survival. So and generally, if you see, it is not possible, for example, to cook the food or to to maintain the appropriate nutrients that are there, that is required for the growth. So generally in the space applications, single cell proteins are used as a food additives. Okay, so generally they are added as a food additives because they have the very appropriate concentration of the carbohydrates and proteins that is required for the growth of required for the growth of, required for the growth, and as well as required for the other nutrient elements that are required for the proper growth. So generally we have, in the space applications, we use these single cell proteins, and we will see how they, they provide appropriate nutrients. So let us look into, before we go into the single cell proteins, like what are these, basically these type of microorganisms. Let us briefly look into, the basic introduction of these SCPs. So these SCPs, if you see, they are nothing but they are some sort of a microbial cells. But those microbial cells, they have, are one of the important thing is like the microbial cells are a different type. Like, it can be a bacteria, it can be yeast, it can be algae, and so on. But the one of the important characteristics of these SCPs or single cell proteins is that it has to be a pure microbial culture, or basically a monoculture. Means it should be a particular one type of either, the bacteria or the yeast, and they should have a very high, means the main major content of its of that cell should be very high in the protein. Okay, and this particular protein is being extracted and is used as a supplement for, for the humans or the animal application, as a food additives. So this is the characteristic of, these SCPs, that it has to be a monoculture. That means it is basically a single type of a bacteria or the fungi or the yeast. Okay, and they are the major fraction of that, of the single cells. They contain a very high amount of the protein concentrations. Okay, and those protein concentrations are suitable for the growth of the, or they are used as those proteins are extracted and they are used as a food additives. So generally, if you see this SCP, single cell protein, they are categorized into two forms. One is called as a food grade, okay, another is called as a feed grade. So what is the two difference? So food grade, basically when you are, means when you are culturing these SCPs, and the proteins that are extracted are suitable for the, the human food applications, then they are classified as a food grade. And if it is not suitable for the human consumption, but it's can be used as an animal feed source, then it is called as a feed grade. Okay, and if you see the single cell protein, now what is the generally means, which microorganisms can be classified as these SCPs? Only those microorganisms which is having a high protein content. So high protein content, the technically it should be about more than 60 to 80% of the dry cell weight. So the if you have a single cell, that means that almost 60 to 80% of that single cell should contain, for example, the protein. Then only it can be classified as under the category of the SCPs. Now, what are the SCPs? So proteins are there, and we know that proteins are made up of amino acids. Okay, and so there can be, the different types of amino acids that are there. Okay, so generally there are some amino acids which can be produced by the humans, some cannot be. So so what type of amino acids that are being supplemented by this, by the single cell protein is also very much important. Along with that proteins, we know that carbohydrates, fats, and other ingredients are very much important. Okay, so so if you look into these SCPs, not only they have a very high, obviously they have a very high protein content, but along with that, they also consist of, the fats, the carbohydrates, and the ash ingredients, water, and other elements, such as, the phosphorus and the potassium and so on. So this makes it, the single cell protein as a, itself a particular meal. And generally, if you see in the, as a food additive, so generally to make sure that, whatever the food that we are taking, they have the adequate nutritional value. We generally add the single cell proteins into it, and especially as I told you in the space applications, where, it is very difficult to carry very high volume or high amount or high quantity of the food materials, or for example preparation of that. So for that's why, for the space applications, generally astronauts takes, the single cell proteins, because in that single cell proteins, generally in a smaller volume and the smaller quantity, you get to you get to get you get the higher concentration of the protein, carbohydrates, fats that are required for the metabolism of the say of a human body. Okay, so that's why these SCPs are widely used. So if you look into the advantage, like as I told you, like if you see the compare, means how do we compare the quantity of this protein? So it can be compared by this one of the example, like on the protein producing capabilities of a 250 kg cow, okay, and the 250 gram of the microorganisms. So the amount of protein that can be produced by that we can obtain from the milk product, that we get it from the 250 kg cow, is it can be means the amount of protein content or the other carbohydrates or other contents, it can is comparable, like how much the protein that we can get from example the milk products of a 250 kg cow, that we can get it from the 250 grams of that microorganisms of this type of single cell protein. So you can see that how much that means high quantity of these nutritional values of this type of SCPs. Okay, and if you see the theoretical studies, so more than the microorganisms can actually produce about 20 to 25 tons of this protein when if they are grown under very ideal conditions. Okay, so that is why we know that these SCPs are widely used. However, still their utilization or their applications in our daily life is has been limited. But especially in the state type of high end applications such as in the space missions and space applications, we are using these SCPs because it can improve the nutritional value, and it will reduce the volume of the food that needs to be consumed, or that needs to be taken up in the space. So, okay, so that's why the SCPs are widely used. Now if you look into some of the advantages of using the microorganisms for producing the SCPs is that the microorganisms grow at a very rapid rate. Okay, and if it is cultured under the optimal conditions. So if you have a bioreactor where you can maintain the different conditions such as the temperature, the pH, the ionic strength, and so on, the microbes, the rate of the growth of the microorganisms of is very high. And for example, in some microbes, they can double their mass in less than 30 minutes. So you can see that that so fast you can produce this type of SCPs. Also, the quality and the quantity of the protein. So generally, all the protein contents in these microorganisms is also very good, or very high quality and quantity of the proteins are produced compared to the higher plants and the animals. Okay, so generally our body has to, so higher plants and animals also contains proteins, but they are mixed with the other things that are there. Okay, and if you see the time frame that is required for the growth of both the plants and animals, compared to that, these SCPs can easily grow in a shorter period of time. Okay, and so generally SCPs are also thought to be if for example in the future we have a food scarcity. Okay, because with the growing populations we will always have a and with the lesser land we will have the food scarcity in near future for sure. So SCPs can actually be used to supplement the issues regarding the food scarcity. Okay, and because in the very smaller volume you will get very high nutrition values or the high protein content. Then there are also the wide range of raw materials, generally using which you can actually produce the SCPs. Okay, so we will see what are those raw materials. So generally, some of the raw materials or the the chemicals, for example, the methanol or some other derivatives, some of the lignocellulosic waste that are generally thrown away, they can actually be used to produce these single cell proteins. And so that means you can use convert the waste or the low value product into the high end or the high value products that we have already discussed. Also, in this case, the SAPs, the culture conditions and the fermentation process are very simple. And since microorganisms can also be easily handled and subjected to the genetic manipulation. So, microorganisms, if you want to produce some particular type of the SCPs, which like with optimized concentration of the protein and the carbohydrates, then can actually be produced by tuning the fermentation process conditions, as well as the carrying out the genetic engineering of these particular microorganisms. But you can see that however there are many challenges in this. So there are challenges of like ethical challenges using a genetic engineered microorganisms as a food derivative. and also like the utilization of the microorganisms directly as a food. So because of these issues, like the commercial scale production or the application in the wider in the wide range has not been carried out or done till date in the SCPs. So this is there are many SCP based products that are already out in the market, and this is one of the so euglena is the name which suggests which is the name of a uh, which is the name of a algae. Okay, and here you can see it is written as the it is a algae. Okay, and this algae can actually produce very high or rich in the protein content. They also have very high protein content of almost 60 to 70%. And this is a product which is called as a Aglena cookie. Means that a cookie or biscuit is made up of using the Aglena algae sources. Okay, and this is one of the area of my research work where we have been working. To use this, we were growing the algae in a bioreactor, and we were making sure that we provide the optimum fermentation conditions and the culture conditions so that we get these euglena which which contains very high rich source in the protein. So as I told you, you cannot directly consume these SCPs as a food. It has to be added as an additive. Okay, and these SCPs were added using in these cookies, so that and it was found that we can improve the nutritional value, okay, of a cookie, especially the fat content, carbohydrate content, the presence of the protein, and so on. And a product was developed. Okay, and in the space application, especially in the JAXA, they are using this type of cookie as a food additives. Okay, so this was one of the project basically where I was involved in production of this type of SCPs. However, there are other products like we have here the spirulina. Okay, so where people use this this type of SCPs if they have they want to fulfill the supplement of the protein in their body. So talking of the substrate that we were talking of, like a wide range of raw materials can be used for the SCP production. So let us look into what are the the substrates that are there. So if you see the important substrates, they are like the molasses, generally that we obtain from as a waste from the sugar industries. Then we have the spent sulfide liquor. So this particular waste is what we obtain it from the paper and pulp industries. Generally paper and pulp. So like the paper that we are using in the notebooks or copies, for example. Okay, so they are being processed through a method of sulfide pulping process, and it generates a lot of waste, which is called as a spent sulfide liquor, because your paper is your note pages for the notebook are nothing but they are made up of the cellulose. Okay, and when you are doing the sulfide treatment, the cellulose breaks and produces some of the glucose derivatives. So those glucose derivatives are present in the spent liquor, and that can be used for the production of these type of SCPs. Then in the wood processing industries, some acid hydrolysis process are taken place, and those waste. So similarly, if you have, so wood is nothing but it's also the backbone is cellulose, and if you do the hydrolysis using acids, so again it breaks down into those type of the glucose derivatives. Okay, and those glucose derivatives can actually be used. Generally, this type of waste are thrown away, and they are also cause the pollution of like water pollution, land pollution, and so on. But these wastes can be used for the production of these type of SCPs. Similarly comes the agricultural waste from different sources, from dairy industry, fruit juice, or the citrus filled hydrosylates, or any other source agricultural waste hydrosylates. Generally they will be rich in the glucose derivatives, so those glucose derivatives can be used for the SCP production. However, there are other some uncommon source that makes it very interesting, for the SCP production. It's like the methane, methanol, and the ethanol. So generally, methane and methanol. So methane is also is some of the one of the the gas that we that are responsible for the global warming. So gaseous methane can actually be used for the production of the SCPs. We'll look into some of the examples for that, and similarly the methanol. So methanol is also waste product generated by many chemical industries. And methanol has can means generally compared to the ethanol, the methanol has a similarly less application. So those and also it is a very low value product that is produced. So both methane and methanol can, if they are used, and they can help in the utilization of the waste, and they become a very cheap on source of the carbon source of the feed for the production of SCPs. Some of the other, hydrocarbon based oils like the paraffin, gas oils, and other alkanes, and they are widely used. So, what is the how means how come they become advantages for us? So, some of the the oil wells or the oil industries, they have very small means they generate a lot of wastewater in which this type of oil derivatives or the alkanes are present at a very low concentration. So those microorganisms can actually utilize those alkanes or the hydrocarbons as a carbon source for the production of the SCPs. So this what we have talked about, are some of the advantages, and these are the substrates by using which the SCPs can be produced. So, but along with the advantages, there are the several disadvantages also. So what are the disadvantages? We'll let us look into that, because so if you see the microorganisms, be it a bacteria, yeast, fungi, generally these SCPs have, as I told, they have high protein content. So the protein content is very high, like it's from 50 to 18%. And means generally anything which is at a very higher values, means everything in the biological system has to be under a particular threshold concentration. So if you are, for example, consuming the carbohydrates, proteins, or fats, or any other thing, so you have to consume it means consumption should be until a threshold limit. Okay, if something is present, either of the carbohydrates is present higher or your protein is present higher, both of them, if one of them is a very high concentration, that is going to affect your body. Okay, so SCPs, as I told you, they have rich in very high protein content compared to the other elements like the carbohydrates, fats, and so on. So because of that, there can be there are some of the disadvantage. So the disadvantage is that if if you have this very high nucleic acid content or high the protein content, it is detrimental to the human beings, because it increases the uric acid level in the blood. So similarly, if you have, for example, the protein rich food, like if you have the lot of quantity of the dals, and which are very highly rich in protein, so they sometimes leak the increase in the uric acid level, and they may cause some of the uric acid poisoning or gut problem and so on. So so generally, when you are producing the SCPs, and when like adding it into any food products, we have to make sure that the protein concentration should not be very high. It should be within the threshold limit. Also, if you see, most of these bacteria cells, they have very the small size, and generally they have a low density. And that's actually, and as you can see that the fermentation process is overall means it requires several components of the medium. So there requires several media components, along with these carbon sources that we have discussed. There will be other media components that will be required for its growth. So generally it makes and then we have to separate out those single cell proteins from this other media components, and then we have to utilize those proteins. So what it does is it makes overall process very much difficult and costly. So these are the two technical disadvantages, and another social disadvantage is like, because we generally think like the bacteria are harmful, and it may lead to some diseases if you consume those type of products. And because of that thinking only, though now we have technologies that can easily produce these SCPs at a comparatively lower cost, but their demand has not been so high till date, because of this the social problems, where people think like the bacteria are always harmful and they may produce some sort of diseases, which is not, because there are some bacteria that can produce very high protein content, and that can be used for as a food supplement. So if you look into these type of SCPs, as I told you, it can be a bacteria, it can be a yeast, it can be a fungi, and so on. So generally, let us look into, very briefly, what are the bacteria, and what are uh, uh, what are their, advantages and the disadvantages, and so on. So hello, am I audible to everyone?
Yes, sir.
Hello. Okay. So let us look into those examples. So one of the important bacteria which is used for the production of these SCPs are the first is, the Methylophilus methylotropous. Okay, so this Methylophilus methylotropous, they have very high reach in content of, this protein, which is almost greater than 70%. And what is the important characteristic of this bacterium is that they can utilize the methanol as, the carbon source. Okay, so generally, methanol or any other, the alkanes or the N paraffin, and like methane gas. Okay, so they can utilize this type of carbon source and convert this particular carbon source into the means they will consume those carbon source, and they will produce the protein within the cells. Okay, and then this protein are extracted out and then being utilized. So so as I told you, so if any, if any microorganisms can use this type of methane gas, N paraffin and alkanes, so generally they are means present as a waste product or in the waste streams of, the refineries or the oil industries. Okay, and methanol is also generally mostly generated as a byproduct by many industries. If this type of compounds can be converted into such value added SCPs, it has several advantages. Okay, so similarly, so because what are the merits of, the using the ethanol for the productions of SCPs are like, so because if you have a methanol, so if we so we have seen of different examples, right? We have the molasses, these things. So until the from first to the fourth, we can see they are, all agricultural based, and there is a huge fluctuations in, the compositions, depending upon the season, depending upon the source from where you are getting these hydroxylates, and so on. So these fluctuations always leads to, difference in the productivity, and you need to add, the other components accordingly. So we have to carry out the chemical composition studies, and then only, we add other ingredients, so that we have all the components that is required for the growth of these SCPs. But coming to this type of, the chemical compounds, especially the methane, methanol, and, these other N paraffin and other alkanes, the advantage is that the composition is, the independent of any seasonal fluctuation. So methanol being methanol itself, and there is no other additives that are there. Okay, and also the other thing is there is no possible source of toxicity in the methanol. Okay, and the methanol also dissolves very easily in the aqueous phase in all concentrations. So, if you look into the agricultural wastes, for example, this type of molasses, hydroxylates, we have to do several processings, like we have seen in the case of the glycerol, that it has to be enzymatically modified, yeah, to convert the cellulose into the glucose derivatives, and the glucose derivatives then being used at the fermentation. But in this case, methanol. So here so there are several pre-processing steps that need to be there, and there are after we obtain the glucose from this derivative, there are some waste, agricultural waste, that are present, like a solid waste, that need to be removed. But in the case of the methanol, such those type of additional steps are not required. Okay, and directly the methanol dissolves in the aqueous phase, and it can be used as for the fermentations. And also, like I discussed, that no residue of carbon source remains in the harvested biomass. So in the case of the biomass, generally, as I told you, like after you extract, the glucose derivatives, you have some solid phase that remains. Okay, and that needs to be filtered and removed out. But in the case of this methanol carbon source, and there is no presence of such type of, the residue. So that's why the utilization of the the methanol as a carbon source by the methylophilus methylotrophus actually makes it advantageous for the production of this type of SCPs. So, along with this particular bacteria, there are several yeast that is also used, and yeast has been the widely accepted and used microorganisms for the the production of the SCPs. And why? Because if you see the yeast, like basically we have studied about the baker's yeast. So yeast has already been used in the as a food additive. So it has make us very socially comfortable for us to utilize the yeast as these SCP proteins and then carry out the consumption of it. Okay, so that's why till date the SCPs has been consumed, means it is most widely accepted, and till date it has been most widely used for the SCP production. Some of the advantages are like they have a larger size, they're easier to harvest or easier to produce, and also they contain the low nucleic acid content. That is very much important. Like in the earlier case for the bacteria, we have very high content of the nucleic acids and the proteins. Okay, so, however, this nucleic acid, because our body cannot tolerate, the human system body system cannot tolerate very high concentration of the nucleic acid, and if it is there, then it may lead to the problems like we already discussed, the uric acid problem and so on. So, it have relatively lower nucleic acid content, a high amino acid content like lysine, and grow ability to grow at the acidic pH. Okay, so these are the some of the few advantages of the yeast fermentation, which makes it suitable for the SCP production. And the micro yeast which are being widely used is the Candida utilis, the Candida liplotica, and the Saccharomyces cerevisiae. So these are the three the yeast that has been widely used for the SCP production. So, however, there remains some disadvantages using this SCP production, using the yeast for the SCP. For the single cell protein production is that they have a lower growth rate. So because of the lower growth rate, the time frame for producing the SCPs are higher, and these SCPs generally have low protein content from the 45% to 65%, and the lower methionine content, like the amino acid, one of the amino acid content is low. So these are the some of the disadvantages that are also there along when carry out use the yeast for the SCP production. Along with that, one of the another advantage is it can use the pentose and the hexose sugar. So as I told you, if you have the agricultural biomass and you enzymatically hydrolyze them, or you hydrolyze them, you will always get a mixture of sugars. It can be hexose, pentose, and so on. So if you would like to have the higher conversion of the carbon source to your final product, and your microorganisms would be able to utilize all the carbon source, be it the 5 carbon source or the 6 carbon source that are 6 carbon source that is present. So in this case, the yeast can consume both of them. So it makes it the higher. It makes it ideal that there is a possibility of getting the higher productivity if both the sugars are combined. So similarly, along like the yeast, the fungi is and the fungus is also one of the SCPs that has been used, and generally the fungi, some of the fungi like the fusarium, okay, or the Phaneroarchaea chrysosporium. So they grow, and they are some of the the single cell derivative of this fungus, and fungus generally grow in form of a multicellular structure, which makes themselves into a mushroom like structure, which we will just see in our upcoming slide. But they generally they use however so like in the earlier cases where we have seen that they can use different uncommon substrate like the methanol, paraffin, or other agriculture derivative. But for the fungus they need to have the simple carbohydrates, okay, that actually produces SCP based protein, which is called as microprotein. Okay, and that is what it is suitable for the human consumption. And if you see, so this is one of the example, like the Phaneroarchaea cryosporium. So So generally they grow on the so if you have ever seen on the stems of the on the barks of the trees, generally you will find that there is a formation of the growth of of the mushrooms. Basically, and they grow on on this type of on the moist environment, on the tree wood, on the bark of the or bark or the stem of these trees. And they generally grow because they are known to be a lignin degrader. So generally, if you see the wood bark, we have already talked about, it is made up of the cellulose, it is made up of the hemicellulose, and the other component that is present is called as a lignin. Okay, so lignin is nothing but it's a aromatic compound, and this type of fungi can actually grow on this lignin, degrade that lignin, okay, and use it for this type of SCV productions. Okay, so this mushroom, so mushrooms basically are of different types and different classifications. Okay, not all mushrooms, only those mushrooms in this particular class we will only study those mushrooms which are the edible in nature. Okay, and so there are different classes of mushrooms and different species and varieties of them, but we will be focused on this particular mushroom, which is the is a particular class of the family of the mushroom, which is called known as the basidiomycetes. Okay, because they are some of the known edible edible form of the mushrooms that are there. So because there are different type of mushrooms, some are edible, some are non-edible, and also some are poisonous. So only those edible mushrooms is what our target of interest. And the basidiomys are the family of this, under this family they are mostly edible mushrooms, and they grow on this wood and the straws. Okay, however, so this growth of the wood and the straws are in our natural conditions, but to to meet the the requirement of the mushroom, especially we are seeing that there has been huge demand of this mushroom because they have very high nutritional value, right? So so to grow under the artificial conditions, especially in a bioreactor system, we need to grow this type of mushroom so that it can fulfill the demand or the food requirements that are there. Okay, so we will look into how this growth of the how the growth of the cultivation of the mushrooms taken are carried out, okay, in our reactor systems. So before that, like as I told you, there these mushrooms are nothing but they are fungi. Okay, so generally we have talked about until now this the yeast, the bacteria, and this particular fungi. This is the Phanerotum chirosporium. So these are all the SCPs. Okay, so that means they are present in a form of single cells, and they each of the individual cells are very much rich and have a high content of the protein. Okay, but in the case of the when we move from the single cell proteins to the mushrooms, one thing that you need to need to keep in mind, the mushrooms are fungi, but they are multicellular. Okay, so the whatever the the fungi mushrooms that you see, they are made up of lot of fungi in together. Okay, and and if you see the edible mushrooms, these edible mushrooms generally belong to the classes of, as I told you, is basidiomycetes, and some of some of the particular species like Agaricus, Auricularia, Tremula, and another class of the asomycetes, mostly the Morchella species or the tuber species. So these are some of these category of the edible mushrooms that are there. Okay, so as I told you, there are almost 4,000 species of this particular family of basidiomycetes, and out of which only 200 are edible. Okay, and the rest are the non-edible, or they have some toxicity, or some are poisonous also. So here you can see that whatever our target will be is to grow such type of 200 different edible varieties of the mushrooms, and make sure that they are very much high in nutrition and protein source and so on. So if you look into the general scheme for the mushroom cultivation, so generally it comprises of the following steps. So let us so we have seen that these are it's a multicellular type of fungi, the mushrooms are, and it can be of different species and so on. But how do we grow those mushrooms? So obviously whatever we have seen until now, we have mostly talked about or studied about the biochemicals, starting from the glycerol, acetone, butanol, ethanol, and so on. So we used to use the bacteria or fungi or the yeast, okay, and we used to put it under a fermented, and then that whatever the biochemicals are there, that is being produced and cultivated out, or produced, and then we do the downstream process to extract out. But here it is a bit different. Okay, so here we are going to grow, okay, the mushrooms within a fermentation system on the fermenter. So also we have to understand how the we have seen that in the case of the bacteria, they have a typical growth curve. Okay, and each growth curve have the lag phase, the exponential phase, the stationary phase, and then finally lag phase or the death phase. Okay. However, the mushroom cultivation is a bit different. The growth of the mushrooms and the nutrient source from which they are taking the carbon source for their growth is actually basically different. So based on that, okay, so this like this particular diagram shows the different different types of substrate onto which we can grow the mushrooms. So we may grow it on a on a tray or on a tray culture, okay, or you can grow it on a stem culture or on the wood logs. On the wood logs, this type of wood logs can be present and can be grown to, which is called a world culture. We can go it in a column culture. We can go and grow in a back culture or the mount culture and so on. So there are different ways or techniques how we can grow that. So let us look into briefly what are the the basic steps. So one of the first step is to selection of a suitable place and types of mushroom to be cultivated. Okay, so as I told you, there are different types of mushroom, and each and every mushrooms have will have their typical conditions that needs to be there. It has to be supplemented with the different the media components. Okay, and then we have to second is to design and layout of your mushroom farm structure. So what is this mushroom farm structure? And so as I've shown you in this diagram, like we can grow the mushrooms on the wall, or we can put it on a plate or a tray culture. And here you can see that all the mushrooms have grown on a tray, and this tray contains the media components which are required for the growth of this type of mushrooms. It can grow in form of a back culture or a mount culture, or in form of a column. So we have to select the architecture or the structure of this mushroom form. Okay, so once you have selected the suitable place and the type of the mushroom, next you have to design and carry out the layout of this mushroom forward structure. And after that, we have to optimize the techniques of compost and the composting process. Now, what is a compost? The compost is so, like for example, until now we have made the growth media components, right? So in this case, for the growth of the mushrooms, you have to provide the adequate, on like in the case of tray culture, they are growing on a particular soil or the soil derivative, or for example, in the tray culture, they are growing on a wood block or other derivatives that are present in the case of the bag. They are those nutrients are kept in the bag, and then on the periphery of those bag, this there's a growth of these mushrooms. So in this case here, you can see that whatever the growth media components that you are going to add, that is added in some different way. All right. So those compost, those type of soil, so generally they have soil type of media in which different additives are added actually. So so we have to prepare a ideal compost, and you have to find out a composting process which should be suitable for the production of the mushrooms. And then are there are techniques or methods of the spawn production and on the spawning. So what is the meaning of the the spawn? So this is a new term. So spawn is a term, if you look in the definition, it is used for the mushroom inoculum containing spores and all small species of the fruiting body. So generally, if you, for example, what does it means? It means like generally, if you see the structure of a mushroom, it has in this type of umbrella and with the stem and so on. However, to make sure they are growing, on this. So they produce lot of or dozens of the spores that are there. So if you start with if you have, for example, for the fermentation of or for the production of the mushrooms, you have prepared your media, for example, which contains all the nutrients that are there. You have sterilized those media, okay, and then you put it on, for example, onto a particular tray, right? And after so this tray is now containing it's a containing the growth media, this the soil, which is nothing but the growth media component, okay, for the fermentation. And on this you have to inoculate. So like we have seen in the case of the fermentation, we used a very small volume of the inoculation, like 10% volume of pre-culture, and then we carry out the fermentation. So similarly, when we are carrying out the growth of the mushrooms, we have to provide the small amount of spores, which can be released also, which can be taken from the previous previous culture of this type of mushrooms, and then it can be added to that particular soil. Okay, so this overall process of taking out the spores and I means you inoculating it into the soil is what is known as the process is called as a spawning. Okay, and after that you have to do the casing and the production of crops. So that means after you have spawned or you have inoculated with the pores, then what will happen, the growth of the mushrooms will going to take place, okay, and the production of that particular crop will carry on. Then we have to carry out the environmental crop management, and followed by the harvesting and the marketing of the mushroom. So so this is a very simple method how it is producing. So we have inoculated it, and then we produce the microbes. These mushrooms are produced, and then they are taken out, and again it is seeded with another inoculation, and then it is carried, it's grown again. So generally, if you see the time frame for the growth, generally it we can, using the single carbon source, we can grow it for 3 4 4 crops, okay. And it is and after the means this is means we can grow it for 3 to 4 crops. That means we can grow the mushrooms for 3 to 4 times, because the soils, they have the adequate the adequate concentration of the different derivatives that can be used for the 3 to 4 times. Okay, so generally, if you see, look into the means the growing condition after you have carried out the spawning or the inoculation, the optimal conditions that are required for the growth of these mushrooms are, it should be water regularity to, and the humidity should be present at 70 to 80%. Okay, and the ideal temperature is about 15 degrees, and pH is about 7, and it almost takes, under control condition, 7 to 10 days for each crop of the mushroom production. Okay, and once and this step can carried out for 3 to 4 crops. Okay, for 3 to 4 cycles to produce more mushrooms. However, one of the disadvantage is that mushrooms generally have a very short life. Okay, generally it's 8 to 12 hours, so they should be stored at proper aseptic conditions under low temperature, and they should be packaged well, so that they do not get rotten very easily. So these are the very basic steps of the cultivations, and how it can be actually cultivated, and the particular the growth conditions. And then, so this actually this particular flow chart is basically summarizing what we have discussed till now. It shows how we can use different steps to produce the mushrooms. So we will keep our discussions until now, and in the next class we will discuss the growth behavior or growth pattern of the mushrooms, how they are growing, and then we will look into some of the advantages of this type of edible mushrooms biotechnology, and we'll look into the different examples, like what are the different type of mushrooms, and how they are grown, and what are the different type of substrates that are used. Okay, so if anyone has any questions or doubts until now, anyone? Hello, hello, am I audible?
Yes, sir, you are audible.
Yes, sir.
Okay, so if not, then let us continue this part in the next class, and we will also have the quiz in the on tomorrow's class. Okay, so tomorrow's class quiz, we will have the remaining part of the mushroom. part, and the glycerol and acetone, that AB fermentation that we have studied. So these three parts will be there for the tomorrow's quiz. Okay. Okay then. So thank you everyone.
Thank you